I am going to be talking, it's a big change from the last speaker, because I'm going to be talking about the virus, and I'm not going to be talking so much about MECSF, but I am going to be talking about this group with the idea of discussing how this type of virus can have this long-term infection. I particularly work on the group B Coxsackie viruses. It's one group of human enteroviruses, but I want to emphasize that there are a lot of different serotypes of enteroviruses, and these are very common viral infections. You find this if you were to take any population and you sampled that population to see if they had these infections. In every population, you would find people who had an enterovirus infection. However, symptoms vary enormously. To tell you a little bit about the virus itself, there are 6 different serotypes of the particular subgroup I work on, the Coxsackie B viruses, and these are viruses that basically get into a cell, and the genome itself is all that's required to end up replicating new virus. And it has a single large protein that it produces. That protein gets processed by viral proteases, things that chew up the virus protein into the smaller pieces. And that's important because probably a lot of the effects of this virus are due to expression of those proteases in the infected cells. The receptor, the protein that actually allows the virus to get into cells, is the Coxsackie and Adenovirus receptor. It's important to understand this receptor because it's expressed on a lot of different cell types, but it's especially expressed on muscle cells. And the disease that I have studied with this virus is an infection of the heart in myocarditis and cardiomyopathy, and this particular receptor is expressed on that tissue, and that's why the virus is found in those cells. However, although my work has centered on myocarditis and cardiomyopathy, Coxsackie B viruses cause a wide variety of diseases, including polymyositis, and they do have this association with MECFS. Part of the reason that they have this wide spectrum of disease is because there are a lot of different cell types that can be infected by these viruses. And of course, again, they're very common viruses. Probably the most common symptom that people have with these infections is flu-like illness. And so they're rarely diagnosed at the acute infection if you're just having flu-like illness to start with. We studied these viruses in mice, and the reason is that we were very fortunate. Compared to a lot of viruses, there's not always a nice model for investigating the disease. But mice happen to have the same receptor that human beings have for these viruses. And so if you give them the virus, they will get a disease that is very similar to human beings, especially if you're using an inbred mouse line that is prone to getting that disease. And when we look at how the disease progresses in these animals, we find that early on, acutely, you have a great deal of virus produced. There is a clearance generally between day 7 and day 21 of virus that can be easily detected. And it depends, that clearance depends on the virus strain and the mouse strain and how old it is. The reason the virus is cleared is because in a normal immune system, you're going to produce antibodies, and some of those antibodies will be neutralizing. They'll basically inactivate the virus. And that neutralizing antibody response is very important with enterovirus infections for actually clearing the infection. However, there is a disease that goes on after that virus clearance in chronic and late-stage myocarditis, and it's characterized by having autoimmune responses against myocardial antigens, having an inflammatory cell-mediated immune response so that your immune system begins attacking the myocardium. And there's also fibrosis, that you have scarring, basically, of the muscle tissue. Now, this stage was considered to be a post-viral stage. The virus was not considered to be present. The work that I have done is basically demonstrated that during this period of time, when you have this post-viral syndrome, you're actually having the virus present, but it has evolved to a different form of the virus, one that is not so readily detectable. So what we have found is that not only are the Coxsackie B viruses associated with this disease, but that it can be detected at the chronic stage of the disease despite the antiviral immunity. However, it's very difficult to culture this virus. And normal assays, you take the virus, you put it on a susceptible cell line, and you see that cells are killed by the virus. We don't find that at this stage, or at least the cell killing is a very delayed and minimal kind. And the argument had been that at this stage you did not have a virus infection because you couldn't detect it using the normal assays. However, our work has demonstrated that the viral genome is present, that virus particles are produced, but the normal assays are not adequate for detecting the virus. Now, since I'm not really talking about ME/CSF, what I'm really talking about is this other disease, myocarditis, chronic myocarditis, and cardiomyopathy, where we have found the mechanism for this virus persistence. And I would suggest that this very well-studied persistent enterovirus infection should be illuminating for ME/CSF, in which enterovirus infections have been noted, that there is this type of phenomena with this group of viruses, that you have an evolution to a defective form of the virus that's very difficult for the normal immune system to clear. It is very dependent upon having this acute infection that actually seeds the body with these defective enteroviruses. Okay, so the reason that we knew that Coxsackie B viruses and indeed enteroviruses in general were a part of the disease of myocarditis was that they had outbreaks of the disease that were associated with outbreaks of the virus. And then a lot of years of study were done using serological evidence of the presence of Coxsackie B viruses in patients with myocarditis. But that's difficult because these are very common viruses. You're going to find serological evidence of infection with these viruses in most individuals if they have achieved, a certain age, because they will have had a chance to be infected by these viruses. Then we very fortunately had the development of molecular techniques that identified the viral genome being present in tissue much more sensitively. And when we did that, we found that about 25% of the patients with this disease actually had enteroviral genomes present in the heart tissue, as compared to a much lower level in the control population. The interesting thing when they did look at these patients, and this is a very nice study from 2001, where they actually took biopsies of the heart, not a simple thing to do, and put those biopsies in culture to detect whether they could see replicating virus. And using the normal assays, they found they could not see the virus despite the viral genome being present. Now, what they were looking for was cell death on culturing. And I will come to why that doesn't actually occur. They also found that people who had this virus present at the time of biopsy were more likely to have more severe symptoms. Okay, we also had a number of studies, and this is not by people in my group, but other laboratories, who looked to see what expression of the virus in this tissue would do if you did not have an acute infection, but actually you inserted the genome in a non-replicating way into the myocytes or into the hearts of mice. And they found that just expression of the 2A protease alone was enough to cause cardiomyopathy in these mice, indicating that even though this virus is not replicating and is not killing the cells, expression of the proteases basically changes the function of that cell, so that eventually the function of the entire heart and functionally it ends up being forced to remodel, ends up causing the disease of cardiomyopathy. Now, these viruses are noted for getting into cells, producing a whole lot of virus, and then releasing the virus by killing the cells. So they're called hit-and-run viruses. And basically, when you infect a human being with these viruses, you're in a situation where you're racing the immune system. People get infected. It's an oral infection usually, or they can get it by wiping their eyes when they've had the virus. But you can't make the human population clean enough to entirely get rid of these viruses. And when they get infected, they have a rapid, acute replication of the virus, primarily, we think, in the gastrointestinal tract. And that has to occur very rapidly because you have a very effective immune response that is mounted against these viruses. So within about 6 to 7 days, you begin to get enough of an immune response to produce antibodies that kill this virus from replicating. So the virus basically gets into as many cells as possible, turns those cells into factories for producing virus, kills them to release the virus, and then it gets excreted and goes on to the next person. So how can you have an infection that would have long-term enough effects on a cell like a cardiomyocyte if it's going to kill the cell? And that was the question. And I know this is one of those horrible molecular biology diagrams, but I just wanted to say that what we're looking at here basically is that the virus gets into the cell, interacting with the receptor for the virus. It binds to that receptor and gets internalized. The genome gets into the cytoplasm, the non-nuclear part of the cell, and just produces viral proteins and viral RNA and ends up inducing a process that breaks the cell open to release the virus. And that is the normal thing that we find with enteroviruses. That includes the whole spectrum from polioviruses to Coxsackie B viruses, Coxsackie A viruses. What we do is to take this virus produced in cell culture, inoculate it into mice, and look and see what happens in the heart of the mouse. And similar to what you find in human beings, during the acute infection, you have a lot of production of virus that can be detected if you infect cells in culture. And the cells in culture that we use are HeLa cells. These are immortalized cell lines. It's an immortalized cell line from a case of human cancer, and they just go on replicating, they go on dividing, they go on dividing. And if you infect an enterovirus in those cells, you kill the cells, and you can see the viruses there because you've killed the cells. And we can see this happening at the early stage of disease, and during this early stage, you actually have in the mouse a lot of necrosis of the heart tissue. You can see that those heart cells in that musculature have actually been killed by the infection with the virus. If you go out to the stage where you have the chronic inflammatory disease with fibrosis and remodeling of the heart, that stage, at about day 28 in the mouse, is one in which virus can no longer be detected by this means, okay? So if you grind up heart cells and you put them into HeLa cells, you don't get a killing of those HeLa cells. We also have another assay, however, RT-PCR, reverse transcriptase polymerase chain reaction, in which we amplify the viral genome so that we can see it, even though there isn't very much of it there. And if you use that technique all the way out, in this particular study to day 53, but in some studies out to day 90 in the mouse, you can see that the virus is continuing to replicate. But at this stage, at the chronic stage, it no longer is killing those HeLa cells. But if you take those HeLa cells and do this molecular assay for the presence of the genome, the virus is there. And we wanted to know why. Why isn't it killing the cells? Because that's something that was never found to occur with enteroviruses. These are very permissive cells. They should be really killed by the virus. And this is the kind of data you get when you do that amplification, that RT-PCR assay. And if you take the wild-type virus, you can see it all the way across the spectrum with primers that are specific for a part of the genome that's inset from the very end of the genome. You can see, using these inset primers, that there's viral RNA in those cultures all the way out to day 18 when the chronic disease starts. But if you use primers that are specific for this one tiny part of the genome at the very 5' end of the genome, you lose the signal. So the virus is there, but it's lost part of the genome. And when we look at these viruses, they've had an incredibly small deletion at a place in the genome that's very hard to assay, right at the very 5' terminus end of the virus. Okay? And that caused a lot of consternation among people in my field. I spent quite a number of years arguing about this because this is considered an essential part of the genome, that you can't possibly lose this part of the genome and continue to replicate. But we were able to show that it does occur. And it's not something that you would really note if you were looking across the spectrum. This has got a fairly small genome. It's 7,400 nucleotides in size. But I'm talking about deletions of only 10 to 49 nucleotides at this very, very end of the genome. And usually the assays for this virus either use an inset part of the genome, or they actually look for the viral proteins being produced by this virus. And all of those are intact. So when you look by these less sensitive assays, you would not know that you were missing this tiny part of the genome. Okay, now I've shown this occurs also in human beings. Now again, this is not, ME/CFS, but this is myocarditis, and this is a patient who died after having a massive heart infection. And because he had such a massive infection, we were easily able to isolate RNA from his heart. And we found again that we had these deletions in his genome. So we were able to prove that this is not just a mouse experiment, that this is something that explains the human data, that you have a non-cytopathic virus infection occurring. Okay, so these tiny deletions, why should we be concerned with them? In having this tiny little RNA structure affected, what's going on? This is a part of the genome that we know is very important for virus replication. It actually allows you to make a lot more of the virus genome. And because it affects positive strand RNA genome, the question was, would you get any viral protein out of these guys? Would you even have enough replication to produce viral proteins? And we were able to show, it takes a lot longer in these HeLa cell cultures to produce the viral proteins, but you can detect them. This is what you get with a wild-type virus. 28 is our wild-type virus. You don't see it at the 24-hour time point because by that time, it's killed all the cells. It's only the defective viruses that go on to produce viral proteins. I think that this is important because the persisting viruses express all the viral proteins. So all of those effects that you might have on the cell can happen, but without actually killing the cells. So this is an explanation of how these persistent infections can remodel a cell, can make it have altered function, but without killing the cell. And if you don't kill the cell, it's far harder for the immune system to clear you out. However, these do produce viral particles. We use neutralizing antibody to show that in these cultures, viral particles were actually being produced that could infect new cells. And the reason that it's important that they do produce viral particles is because you can have an ongoing infection with the virus. Even though it's producing virus at very low levels, it continues to produce those viral particles. And that way you can have a prolonged infection. These viruses also have a characteristic that is noted for persisting enteroviruses. They have an altered positive strand to negative strand ratio. Okay, what I'm talking about here is in this type of virus, the genome itself encodes the viral proteins and is known as the positive strand. There's an anti-genome that just serves as a template to make more of the genome for producing new viral particles. Characteristically, we have found in these persistent enterovirus infections, and this had been noted in this study in 1990, that if you have a persistent enterovirus infection, you have equal amounts or nearly equal amounts of the genome and the anti-genome, the template. And that is, again, something not noted for enteroviruses. And that is something that these particularly deleted enteroviruses have. They produce a lot more of the negative strand than is normal. Okay? We have also found that we can actually replicate this effect in cell culture, but it's not these continuously replicating cancer cells that we can make this in. You actually have to use cultures of pancreatic cardiac cells, and we've actually found a lung epithelial cell line that will do this as well. And the characteristic of these cell cultures is that they stop dividing without cell death when you achieve sufficient cell density. And basically they go more into a state that is characteristic of the tissues that you'd find in muscle and in the myocardium, basically they have differentiated. They're doing what they're doing. They're not continuously dividing. They are very restrictive for wild-type virus. But if you pass wild-type virus in these cell cultures, you find that after a certain period of time, you've lost the very 5' end. You start being able to detect a lot more of the negative strand virus. And I don't have a picture of it here, but if you look at HeLa cell cultures of these viruses, they're again replicating in a non-cytopathic manner, meaning that the old assays would not detect them. And when you look in these cell cultures, markers for cell division indicate that these cell cultures are actually quiescent. Okay, so the part of the genome where we have these deletions is a part that's known to bind host factors. And labs other than mine demonstrated that that part of the genome was not only important for the positive to negative strand ratio when they engineered mutations, and in this particular case they're using poliovirus, but it's also a place where this factor binds, and this factor is very important for having a lot of genomic replication. Okay, our particular virus has lost that binding site for that host factor, and that host factor is one that's it's only in the cytoplasm. I'm sorry, that's a typo. It's only in the nucleus, except during mitosis, except during cell division, or if you actually have a late-stage virus infection. And we've been able to show that factor, although present in these cells, when the cells become quiescent, you lose that factor. Okay, you take a really long overexposure. These particular, that's the HeLa cells, and that's an ovarian cancer cell line. They have a lot of that factor in the cytoplasm. They have a lytic infection with wild-type virus. Our cardiac cell cultures, when you do the same extract and look at what's present outside the nucleus in the cell, you find that they have very little. You can see a little of it coming up if you do a long enough exposure. Okay, and we took our viral RNA from the deleted viruses, and we found that it did not bind this factor at anything more than what you would find with any RNA. So once these deletions have been generated, the wild-type virus genome will not be regenerated because you've lost the binding site for this particular host cell factor that's necessary for a high-level replication. If you treat the cells with it's a silencing RNA, something that will prevent expression of this particular protein, you end up not being able to detect virus replication because it's simply required at some level. Okay, so what we found is basically that there's a host cell factor in the nucleus that has to enter the cytoplasm in order to have a normal type replication. And in tissues like the muscle, that factor is not present. And when you don't have that factor present, you go into a defective form of this virus's replication, one that selects for these deletions. And that selection for deletions ends up causing you not to kill the cells, but to have an internal replication of the virus for a long time. And because in that cell you have the proteases being expressed, you start altering the function of that cell. Okay? And this is kind of a model that had been given for what happens in myocarditis. And this was before we had been able to detect the persisting viral genome. You had an acute myocarditis with a whole lot of production of inflammatory agents, and you had an immune response that would tend to kill a lot of cells in those infected myocardium. You had a longer-lasting cell-mediated immune response that was important, they felt, for viral clearing, but also caused inflammation. Eventually, you would go into fibrosis and remodeling of the heart, but this was thought to happen at a time where there was the absence of the virus. Our work has actually shown that during this period, you actually do have the virus present. Okay? So what we have as our hypothesis is that it's this particular type of cell, a fully differentiated type of cell. I mean, it's quiescent only in terms of cell division. They're actually doing most of the function of your body cells that have already fully differentiated and stop cell division. But that's an environment in which these enteroviruses, if they get into the cell, and it takes an acute infection to get them into the cell, leads to a selection of a defective genome, okay, one that's missing a part of it. And the result of the selection for that defective genome is that you have the ability to infect a cell for a long period of time, affect how that cell actually works, and has a lot of the characteristics found for persisting enteroviruses. So we've provided basically a mechanism, a molecular mechanism, for what had actually been detected for a long time in myocarditis and had also been found in CFS and ME. Replication without rapid cell death is necessary in order to have the long-lasting effects. You actually, in this way, can have a persistence of the infection despite having an immune response. And we know you can actually put these defective viruses into a mouse that has a normal immune response, and that mouse will not be able to clear the infection. So once you generate it, it takes a lot longer to get rid of it. However, it's a dead end for the virus. It's kind of an interesting sideline for the virus because really when you think of the evolution of these viruses, what's important is actually that it go on and infect the next person. But you've had a selection within the patient's body for a virus that can have an effect upon the tissue, can cause a pathological effect. And again, the immune system does not readily clear this type of infection. We're not sure totally why that is. We think it's in part because you have such a low level of production of virus proteins and as well the genome that it's much more difficult for the immune system to hit every infected cell. And because it's a slow replication, there'll be a lot of infected cells that are still at the point where they're not producing enough of the virus to actually be seen by the immune system. So we think it actually has a chance to persist for a lot longer within the tissue than the normal virus infection would, simply because you're producing less virus. Okay? So what does this mean for the disease? We know that a lot of people with ME/CSF have infections of muscle or the gastrointestinal tract. Dr. Chia is going to be speaking after I, and he's done a lot of work showing people are actually excreting virus. But of course, muscle biopsies had actually indicated the presence of enterovirus in these patients as well. Severe acute infections are likely to lead to this type of disease. I don't know, whether whatever leads to ME/CFS is more likely to give you a severe acute infection, or whether the population that has this type of enterovirus infection by chance had a poor immune response, either because of their own genetics or their health situation, but they're more likely to have had a high infective dose originally, leading to this type of infection that selects out the defective viruses. Expression of the CVB proteins in such tissues can generate a chronic inflammation of those tissues. That's to be expected because you've had an acute infection that generated a really good immune response. So having the virus around, even though it's at very low levels and it's not going to kill all the cells, is likely to create a situation that is greater for having a chronic inflammation. And we do see that in the mouse, in the heart, and in polymyositis models in the skeletal muscle. Okay? And I'd like to mention my collaborators in this research. Stephen Tracy has been my collaborator for many years working on this. But we have quite a group of people in the state of Nebraska who are actually looking at this type of chronic infection and also the molecular biology of the viruses. And we're very fortunate that NIH has decided that this particular form of replication of the virus is of interest to them, so we now have funding from NIH for this work. We did have funding from the American Heart Association in the past for looking at this, and we've had a lot of generous support from the families of patients with myocarditis. Okay? And that's what I have to say.